A couple people said they couldn't get into Blackboard yesterday. Apparently Blackboard just decided that everyone's passwords were not good anymore.
Yep.
Okay, it wasn't, I got 2 emails with the same problem, so I figured there was probably more than just those 2 people. There was a quiz that was due today, or it was due at 8. I pushed that deadline on that to 5, so if you weren't able to get into Blackboard yesterday to do the quiz, you can do it today and still get those results. Yeah, I don't know why it suddenly decided everyone's passwords were wrong.
So they only let you change your password when you log in on a campus computer.
Yeah.
So once you actually go to the library log, then you can change it. So until then, it says your password's wrong now.
That's fun. Anyway, I did hear about that, so I did push the deadline for today's quiz to 5 p.m. so you can go in and do that today. Still. Other questions, concerns, issues? All right, cool. So let's finish up with the animal lecture, animal diversity. So we were talking about the Lophotrochozoans on Friday. And we talked about, I believe we talked about annelid worms and mollusks. And I don't think we got the platyhelminthes. Does that sound right to you guys?
Yep.
Okay, cool. So that's where I picked it up then, which is convenient. So the platyhelminthes are also known as the flatworms. Platyhelminthes are acoelomates, meaning they don't have a true coelom. What's a coelom? It's a body cavity, yep. And how is a true coelom different from a pseudocoelom?
True coelom.
And the meso?
Like, it's more defined, like they have the cytoplasm, right?
Sort of.
Different sections of it, right?
Yeah, it doesn't necessarily. I don't necessarily that it has different sections. It's that it's surrounded on all sides by mesoderm tissue. And the pseudocoelom is between the mesoderm and the endoderm. So that's the difference between a true coelom and a pseudocoelom. These guys are acoelomates. What does that mean, to be an acoelomate? They don't have a coelom, exactly. So they don't have a fluid-filled cavity. Flatworms include a number of different types of things. There are some marine flatworms that are super interesting and cool. These guys you might have seen before. That's a planaria. It's a type of freshwater flatworm that is a tridivore. It eats little organic things that are on the bottom of ponds and lakes and things. They're kind of fun. There are also a fair number of parasitic flatworms. This is a liver fluke. Liver flukes are a pretty substantial problem for cows, among other organisms. And they do actually get into the liver and parasitize the liver, which is not great. Those of you who have dogs may have heard of tapeworms. Has your dog ever got a tapeworm? Mine has. Ooh, it's gross. But tapeworms are another parasitic worm. Basically live in the intestine of some animals and feed off of that animal's food. So they're not a great thing to have in your body. But, so some of them, the upshot here is that some of them are free living and some of them are parasitic. Okay, any questions about any of that stuff? All right, so let's move on from the Lophotrochozoans to the Ecdysozoans. All right, so we're kind of in this part of the animal tree here. So we just looked at the Lophotrochozoans. We're now looking at the Ecdysozoans. There's a whole bunch of things in there. I'm really only going to ask you to know about 2 of these groups, the nematodes and the arthropods. Nematodes are roundworms. Nematodes are pseudocoelomates. Nematodes are one of the most abundant animals on the planet. In every tablespoon of soil, there's several thousand nematodes. They're often very tiny and they're just everywhere. Most of them are harmless, free-living organisms, although there are some parasitic nematodes as well. Hookworm, if you've ever heard of hookworm, is a parasitic nematode. So some of them are free-living and some of them are parasitic. Questions about that? All right, so we've got the segmented worms, flatworms, roundworms. 3 different groups of worms. So a little bit confusing, but they're all in different groups. Okay, then the arthropods. The arthropods are, the similarity that all the arthropods have is that they all have a crunchy exoskeleton made of chitin. Arthropods are true coelomates. They have substantial segmentation. So some of these arthropods are things like centipedes and millipedes, which have lots and lots and lots of segments, each with their own little pair of legs. But also the insects are an arthropod, the crustaceans are arthropods, spiders and ticks and things are arthropods. Remember I told you my favorite blood-sucking parasite was the land leech. My least favorite blood-sucking parasite are ticks. And particularly for me, the paralysis tick, which in certain unlucky people can trigger an allergy to mammal meat. So I now no longer can eat any mammals, any cows or pigs or sheep or anything like that, because I'll go into anaphylactic shock. It's really fun. And that allergy was triggered by being bitten by a paralysis tick, which also, as you might guess from the name, can actually paralyze its prey as well. So, least favorite blood-sucking parasite. Let's see, what else do I have to say about arthropods? I think that's about all I have to say about arthropods. Any questions about these guys? No? Okay. All right, that brings us to the deuterostomes. So the ecdysozoans and the lophotrochozoans, those were both groups of protostome. What is the difference between a protostome and a deuterostome? Exactly. So the blastopore, which is the first infolding of the embryo, becomes the mouth in a protostome, and the blastopore becomes the anus in a deuterostome. Okay? The two groups of deuterostomes, well, there's actually technically three groups of deuterostomes that I want you to know. The echinoderms and the chordates are the major groups. Within the chordates, I'd like you to know what the vertebrates are. So we are a vertebrate. We fall within the chordate phylum. So there are some other things in there that are not vertebrates. So for example, sea squirts, and this is called a lancelet. It looks like a fish, but it's not a fish. These guys are in the same phylum with the vertebrates as well. Yes?
So you said we need to know the chordata and then the vertebrates specifically within the chordata and
Correct.
not worry about the others?
You don't have to worry about the other two. I'm not going to ask you questions about those two. I just think, I'm going to tell you a little bit about them because I think they're cool, but that's not a group that I'm going to require you to know. So everything that's in red. Do we have any colorblind people in this class? Can you tell that those are? Okay, cool. I'll try to tone down the use of red in my PowerPoints.
Okay.
So yeah, anything that's in red is what you need to know. All right, so let's take a closer look. The echinoderms. Probably the echinoderm you're most familiar with are sea stars. Marine biologists get very aggravated if you call them starfish, like this slide does. I stole this picture off the internet. Marine biologists are like, it's not a fish, and they get really mad. But so they prefer sea stars. So sea stars are probably the ones you're most familiar with, but there's a bunch of other types of echinoderms, like brittle stars. You guys read a paper about brittle stars in lab. Sea urchins, which are basically little balls of spines, and sea cucumbers and feather stars. My college roommate did research on sea cucumbers when she was an undergraduate, and she named all of her sea cucumbers after friends of hers. And Claire, the one she named after me, got into the water recycling system and escaped to the ocean. And the one she named after my ex-boyfriend puked his guts out and died. So, found that very amusing at the time. But yeah, sea cucumbers are, a lot of these things are filter feeders. So sea cucumbers are a filter feeder, feather stars are a filter feeder. Sea urchins are actually herbivores. They're pretty voracious. Voracious. Voracious is a real word, ferocious is made up. They're pretty voracious grazers of kelp. They can actually do a lot of damage to kelp forests if they get overpopulated. Sea stars, on the other hand, are actually predatory. And they're pretty cool because they're one of the few animals that actually has external digestion. So what they'll do is they'll find a bivalve, a clam, or a mussel, and they've got little mouth parts that can scrape a hole in the shell of the clam or the mussel, and then they'll actually evert their stomach, so they'll turn their stomach inside out, put it through the hole in the mussel, and then digest the mussel on the inside of the shell, and then pull their stomach back through their mouth. Yeah.
Did you say the sea urchins were filter feeders as well?
No, sea urchins are herbivores. Yeah. So, yeah, and where you have a lot of sea stars, you tend to have very few mussels. They tend to keep mussel populations pretty well in check. All right, other questions about these guys? So these guys, echinoderms, are actually one of the phylums that's most closely related to the chordates. The chordate is the phylum to which we belong. We are a chordate. The unifying characteristics of the chordata is that we have a dorsal notochord. And the notochord is a flexible rod-like structure in a vertebrate that is present in the embryo. And then during development, the notochord actually becomes the discs between the vertebrae. So it's still a flexible structure and it permits movement of our discs. As adults, we don't have the full notochord anymore. We only have it as the embryo in the embryonic stage. Some of those other critters that I told you that don't look a lot like vertebrates, like the lancets. The lancets look like a little fish, but they're not a little fish. They're a different thing. But they have the notochord throughout their entire lifespan, so that's what designates them as a chordate. And then these sea squirts, which basically are just a filter feeding sac. As adults, they don't look a lot like the rest of the chordates, but in the larval stage, they look a lot like a lancet. So they have that notochord. They're actually a little swimming thing, and then eventually they settle and they metamorphose into these filter feeding sacs. So the hypothesis for how vertebrates started to evolve from a very simple animal is that vertebrates are basically a larval stage that stopped developing into the adult phase. So it became advantageous to remain in the larval stage and remain more mobile, and then we had evolution from there to all of the different types of vertebrates. So all the vertebrates that include the fishes and the amphibians, reptiles, birds, mammals, including ourselves. Okay, I think that's it. Any questions about animals or animal groupings? Okay, any questions at all for the exam? Yes?
So for the exam, I guess in regards to need to know all of the different, that was my question. How many different phylums are you going to use now? And
Okay, so on the review sheets, I think I put all of the taxa that you need to know.
okay.
Basically, if I put a slide of it in one of my PowerPoints, it's one that I want you to know. There's a lot more in the book that I didn't get to. So if there's not a PowerPoint slide of the group, I'm not going to ask you questions about it. So those are the two places to make sure that you know all the taxa. Look at the review sheets and also look at the PowerPoints. Because there are just a crap ton of different, taxonomic groups that one could memorize. And you will have to, at some point in your biological careers, you have to memorize more of them. When you take zoology, for example, you get to know a lot more animal groups than I'm requiring you to know for this exam. But this is like the basic framework of how to categorize life, and then you can diversify out from there. Other questions? Yes?
Can we go over why bacteria and archaea are different groups?
Yes. What do you guys remember about some of the differences between bacteria and archaea?
Have peptidoglycan. That one?
Yeah, so the protein that makes up the cell walls, the eubacteria have peptidoglycan, the archaea don't. I don't actually really even need you to know the word peptidoglycan. If you can just say that the cell walls are made of different things, that's fine. What else?
They have archaea have similar gene expression to eukaryotes?
That, exactly right. So, there's actually a difference. Hopefully you guys, when you took biology in high school, you remember that you've got your nucleotides that make up the DNA, and they go in codons, or sets of 3, and each set of 3 codes for a particular amino acid. There's actually a couple codons, or sets of 3 nucleotides, that code for a different amino acid in bacteria than they do in the archaea and the eukaryotes. So that transcription process is a little bit different between those 2 groups. So that's exactly right. And then anything else?
DNA replication? Or
Yep, the DNA replication is more similar between archaea and eukaryotes than it is for bacteria. And did you have another one?
Bacteria have horizontal gene transfer.
Actually, both bacteria and archaea have horizontal gene transfer. So all the prokaryotes can do horizontal gene transfer.
Is there any that we should know other than that? That was all I saw on the slides.
Those
but-
were the 3 that I think I said. I don't remember. Oh no, there's one about the cell membranes. The lipids in the cell membranes are also different.
Right.
Yeah. So that's the 4th one. Yeah.
So the second one is difference in transcription or difference in,
Transcription. But there's also differences in DNA replication.
that's the one.
Yeah. Did that help answer your question? Yeah, so the fact that the archaea have many more similarities with eukaryotes than they do with bacteria means that if you draw a tree of the domains of life, so a domain is our highest taxonomic group that we have, that we've named. All of the taxonomic groups, this is one thing you have to remember about taxonomy. All taxonomic groups are names that were made up by people. In a perfect world, those names actually reflect evolutionary history. How well they do that is up for debate. But we're pretty confident about the domains. And if you build a phylogenetic tree of the 3 domains, you have the eubacteria over here, more distantly related. And then you have the archaea, and then you have the eukaryotes. So the archaea is more closely related to the eukaryotes than either of these are to the eubacteria. So the eubacteria are the most distantly related group. So even though we call these guys together prokaryotes, what kind of a phylogenetic group is that? The paraphyletic group. Because the common ancestor way back here, eukaryotes are also a descendant of that common ancestor, so that would be a paraphyletic group. Okay? Any questions?
I'm trying to find,
Can you go over the paraphyletic, monophyletic?
Polyphyletic thing?
Yeah.
Yeah, okay, so, I'm gonna draw, can I erase this? I'm gonna draw a little bit of a bigger tree.
everybody knows that.
Yeah, no.
It's damn
All right.
good stuff.
So, what's the definition of a monophyletic group? Yeah, common ancestor and all descendants of that ancestor. In any tree, I'll draw another branch. In any tree, you can draw a number of monophyletic groups. So anytime that you include the common ancestor and all descendants of that ancestor, that's a monophyletic group, right? So all of these boxes I just drew are different monophyletic groups. We have the common ancestor and all descendants of that ancestor in each of these cases. Those would all be monophyletic, okay? A paraphyletic group is if you include the common ancestor and some, so here I've excluded this tip. I'm including the common ancestor and some, but not all descendants of that common ancestor. We do have a fair amount of paraphyletic groups in taxonomy, because our naming systems aren't perfect. We don't like to give up names we've already come up with because we're stubborn. And so there's a number of groupings of organisms that are actually paraphyletic. We really try to avoid, though, polyphyletic groups. So if I drew a box here and I said that these two organisms belonged in a group together, that would be a polyphyletic group, because this doesn't even, their common ancestors way the heck down here. And that's not, the common ancestors is not even included in that group, okay? So that's a polyphyletic group. And we in taxonomy, we really try to avoid those. And when we discover that we've accidentally made one, we'll usually rename the group. Because it represents a naming system that does not reflect the evolutionary history of the group. And in taxonomy, that's the whole goal, is to come up with ways to name things that reflect the actual evolutionary history. But because we weren't here millions of years ago when this evolutionary history actually happened, sometimes we're wrong. And sometimes we get new evidence, we have to reevaluate our naming system. Yeah.
I was gonna ask, when is the SLC exercise due?
Oh,
Wednesday, isn't it?
Wednesday, yes, it's due Wednesday. Yeah, and I forgot to put that on the announcement slide, but yes, we do have an SLC exercise. Yes, it is due Wednesday, and it'll help you study for the exam, so go do it. Other questions? Yeah.
Are the prokaryotes just in the ecological, like the benefits or like the function of the prokaryotes in the ecological system? Is that just for like nitrogen fixation?
Well, nitrogen fixation is a really important role of prokaryotes in the ecological system, because they're the only things that can fix nitrogen.
Okay.
We actually, except for humans now have technology that allows us to fix nitrogen, but it's extremely, and it takes a lot of energy for us to do it.
Efficient.
Yeah, the prokaryotes are much better at it than we are.
Yeah.
So that is one really important role of prokaryotes within the ecosystem. Also, they are major decomposers. So they break down dead organic material and return those nutrients back to make them bioavailable for other organisms again.
Okay.
They also they play beneficial roles for a lot of, we have symbiotic relationships with a lot of these microbes as well, animals do. And so they play beneficial roles for animals in terms of digestion in most cases. But also there's prokaryotes that can help prevent you from getting sick from pathogenic prokaryotes. So there's a number of beneficial roles of prokaryotes in the ecosystem.
The one that is understand how membrane-bound organelles may have risen.
Mhm.
Is that like the cyanobacteria and the mitochondria? Is that what it's talking about, or?
Yeah. So it's talking about that endosymbiosis hypothesis, where the mitochondria and the chloroplasts are thought to have previously been free-living prokaryotic organisms that were engulfed by another organism and became those organelles. The other membrane-bound organelle that we talked about the origins of was the nucleus. So what's the difference in the hypothesis for how the nucleus formed versus the mitochondria and the chloroplast?
It was its own thing, right? Or is that what you're talking about?
Well, yeah, how do we think the nucleus formed?
Is it the folding of the endoplasmic reticulum or whatever?
Yeah, it's actually the infolding of the plasma membrane, but yeah. So we think that the plasma membrane around the cell infolded and then eventually surrounded the genetic material and formed the nucleus that way. So it's just a little bit of a different process for that membrane-bound organelle than for the chloroplast and mitochondria.
Yeah.
Yeah. Other questions? Yeah.
How would you compare the patterns of multicellularity and plant cell?
So what are the things that are required to be multicellular?
Adhesion,
Adhesion,
communication between cells,
communication,
what is it, diversity, or not diversity,
you're on the right track. Shoot, I'm blanking on the word too.
differentiation.
Differentiation, thank you. And then the last one,
Cells in the middle don't contact each other, or don't contact the environment, so.
so what do you have to do?
Active transport.
Transport, some sort of transport. So if I'm asking you to compare those patterns, I'm asking you between plants and animals, I kind of want you to think about how multicellular plants and multicellular animals deal with those four things differently. So for example, for cell-to-cell communication, in animals we talked about gap
Oh, yeah.
junctions, and in plants we talked about plasmodesmata, right? Or bulk transport around the body. In animals, you have a circulatory system with a heart that's actually pumping blood around, whereas in plants, you have a vascular system that is moving liquids around the body in a different way, without a heart. So that's the kind of comparison I was looking for there. And you could go back to the PowerPoint slides and kind of look at those.
And like the benefits and costs of multicellularity,
Mhm.
I can't exactly remember those. The like reproduction I know is one,
Mhm.
and then being able to eat other small things is
Yeah.
benefit.
Yep, and also potentially escaping predation is a benefit.
Uh-huh.
Yep, but reproduction, is that a benefit?
That's
or-
a cost.
That's a cost, because not every cell can reproduce. So some cells are giving up reproduction,
Uh-huh.
and potentially reproduction gets a heck of a lot more complicated as well in a multicellular organism than it is in a unicellular organism. If you're unicellular, you reproduce just by dividing yourself in half.
Right.
If you're multicellular, you can't do that.
Is there any other cost to being multicellular?
Um,
Maintaining
hmm?
position. Maintaining position.
What was
Was
it?
that- I think that was a benefit.
Of being
Yeah.
multicellular.
Yeah, I know I had something else on there, I'm trying to remember. I think,
Get access to resources.
yeah, so the other costs are things like transport around the body
Uh-huh.
and accessing the resources, and then competition between individual cells in the body. So I gave cancer as an example of something that is a cost of multicellularity. There's no such thing as cancer in a unicellular organism.
Yeah. Like so the super kingdoms,
Yeah.
am I just gonna have to like memorize that slide and be able to like fill it out, or just know each like characteristic that,
So
Classifies them.
there is a set of, actually, I think I want you to know all of the ones within the eukaryotes. So yeah, you should be able to give examples from each of those super kingdoms of
Uh-huh.
eukaryotes. So, make sure that, and you can use the examples from the slides, because within each super kingdom, I gave you a couple of examples.
mhm.
Yeah, that's basically, I'm not gonna ask you to draw the whole tree.
Right.
That's craziness. But, you should be able to, within the different eukaryotic super kingdoms, be able to give me some examples of some things that are in those ones,
Mhm.
okay? Or if I ask you about a particular organism, you should tell me what super kingdom it belongs to.
Right.
So I'm like, okay, what super kingdom does an animal belong to?
That would be Opsicon.
Yeah, piscicons, exactly.
Then like all the plant stuff that we have to know,
Mhm.
like you may ask us like, what is a conifer? You have to
Yeah.
give like 3 examples.
Yeah, I might be like, what's a conifer? And you say gymnosperm.
Right. Cause like there's gonna be like,
Or what's an example of a gymnosperm? And you could say conifer or
Mhm.
ginkgo or something like that.
okay.
I know it's a lot of taxonomic groups to memorize. This is one of those things where you just gotta memorize it. I suggest flashcards, that helps me, but whatever memorization strategies you guys have. Part of intro bio is learning the vocabulary that allows you to communicate with other biologists. And part of that is just memorization. So, when a biologist comes up to you and says, I study gymnosperms, you have to have some idea of what they're talking about. Or I study X plant, which is a gymnosperm, and you can be like, okay, I can put this into a category in my head that I can make sense out of it and make some deductions about it.
Cause pterophytes and lycophytes, those are tracheophytes.
Yes, both pterophytes and lycophytes fall within the tracheophytes. So, especially within the plants, we have a lot of nested categories.
Uh-huh.
So you've got your land plants, you've got your tracheophytes, and then, you got your seed plants, and then your seed plants are divided into, you could go to your angiosperms, and your angiosperms are divided, and those are all like nested within each other.
Right.
So it's a bit complicated.
Cause, so if you asked me to give examples of tracheophytes, I would do pterophyte or lycophyte.
Or seed plant. Seed plant is
Seed
also a
plant.
tracheophyte.
But if you wanted like one that was, if you just asked me examples of lycophyte, I would like give fern, horsetail, or.
Well,
seed plant.
lycophytes are their own thing. So I wouldn't ask you for an example of a lycophyte.
Right.
Because I only gave you the word lycophyte to describe, well, lycophytes are the club mosses, basically.
Mhm.
Those are the ones that are still alive today, are club mosses. They're the only thing in the lycophytes that are still alive today.
Right.
Yeah, it's a lot, I know. Other questions? You guys wanna do some practice questions?
Yes,
Okay.
yes, I do.
All right.
Photosynthesis.
So, we'll do this one as like, why don't you guys, think about your own answer and write it down, then talk to your neighbor.
Photosynthesis. Means they, yeah, they get their energy from.
All right, so if you're looking at the word photoheterotroph, what does the photo mean? It's getting energy from light. So, not necessarily photosynthetic,
Ooh,
because photosynthetic, what are you doing in photosynthesis?
creating CO2.
You're creating your own organic carbon molecules, right? So this is an organism that can get its energy from light, but this organism is also a heterotroph.
Eats other,
So what does that mean?
it eats other animals, right?
It can't fix
Oh,
carbon.
it can't fix carbon.
So is this organism photosynthetic? No. It can utilize light energy to do things, but it can't fix its own carbon. So what I want you to be able to distinguish is that in these words about metabolisms, these are all describing cellular metabolism. The first part, photo versus chemo, is talking about where organisms get energy. And then auto versus hetero is talking about can they fix carbon, okay? And pretty much all organisms can use chemical bonds to get energy, but not all organisms can use light. And so photo means they can use light energy, yes? Not yet, that'll be more in the plant science. Yeah, if you can, you should know generally what, I mean, cuticle's pretty important for adaptations to living on land, but we're gonna, yeah, we're gonna get into that more when we talk about plants, which is our whole next section of plants. Yeah, all plants all the time. All right, so, going back to this question of metabolism, does that kind of make sense? What those different words are referring to? So, yeah, go ahead.
It was hetero versus what? What's the other one?
Auto.
Auto, okay, thank you.
Yeah, so, hetero versus auto and photo versus chemo. So pretty much all life can get energy from chemical bonds, but not all life can use energy from light, only some can. And so if you can, then you're a photo. If you can't use light and you only can rely on chemicals, then you're a chemo. And then auto versus hetero, if you can fix carbon, you're an autotroph, and if you can't fix carbon, you're a heterotroph. Okay, that make sense?
So the plants that release CO2 into the atmosphere are autotrophs.
The plants that release CO2 into the atmosphere?
That was not, sorry, that was back then. They take in carbon dioxide from us.
Plants are photoautotrophs because they can use light energy and they can fix their own carbon. Yeah, and they do take their carbon dioxide out of the atmosphere. That's where they get their carbon dioxide from, that they fix into their organic compounds. All right, so photoheterotrophs, organisms light energy, but can't fix carbon. What's the only group that's known to contain photoheterotrophs?
Fungi?
Hmm?
It's not fungi, is it?
Nope, can fungi fix carbon? Well, no, can fungi use light?
Prokaryotes.
They do, yeah.
How do they use
I,
light?
I thought they did. I know that you grow them in the dark, but sometimes they take like some light, like 65K light.
I don't think it matters what light you give to fungi because they can't use light energy.
Hmm.
They're all fungi are chemoheterotrophs.
Okay.
Yeah. That's weird. They tell you to put light on fungi to grow them. Maybe they
Yeah.
like the heat.
It's like if you thought like a dark attic,
Uh-huh.
like a dim attic,
Uh-huh.
like that.
Is like a good
Yeah.
place to grow. It probably has to do with they like a lot of moisture generally, and they probably if you like put them under a bright light, it's going to dry them out.
Uh-huh.
So that's probably why you don't want to put them under a bright light.
Prokaryotes, right?
Prokaryotes, right. So prokaryote, okay, so there's a lot of things that are chemoheterotrophs or photoautotrophs. Those two things are pretty common. The things that are rare are photoheterotrophs, so things that can use light energy but can't fix their own carbon, and chemoautotrophs, things that can fix carbon but can't use light energy to do it. The only group that contains photoheterotrophs and chemoautotrophs is the prokaryotes. Okay?
Bacteria, the chemoautotrophs, right?
Yeah,
So
the
bacteria and
the
archaea.
cement bacteria are an example of a chemoautotrope. And there are they're not super common, but there are some photoheterotrophic bacteria that can get, use light from the sun to create energy, chemical energy, but they don't have the capability to take carbon dioxide and turn it into organic molecules. So they have to get their organic molecules from some other system. They could just use some of that light energy
Mhm.
to help power their cells. Make sense? Okay. Any more questions about metabolisms? All right, we got a clicker question. So get out your clickers.
The same process.
Where's my clicker? Did I not start it?
D.
I didn't start it. I'm starting it now.
D.
Give everyone a chance to get their clickers out.
What? It is. Did it work? Nope.
Yeah. All right. Okay, now you can click, so go ahead and click now. You clicked earlier, click again. Definitely more than, okay, there we go. Anybody else need to click in? Everybody clicked in? Okay. All right, so what is transformation?
Isn't that when the cell dies?
Stressful responses, right? Like
Okay,
makes
so
the membrane's more permeable or something like that?
Yeah, exactly, so a prokaryote gets stressed. When it's stressed, its membrane becomes more permeable, and what does it pick up?
Other dead prokaryotes, like their cells, like their DNA or something, and
Mhm.
it tries to like.
Yeah, it picks up little fragments of DNA from the environment, all right? So that's transformation. It's a stress response of prokaryotes, and they can pick up little chunks of DNA from the environment, okay? What's transduction?
When a virus picks up a part of the gene?
Yeah, so that's when a piece of bacterial DNA is transported by a virus to a new prokaryote, right? So they both are examples of what?
Gene.
Exactly, horizontal gene transfer. So it's a way that prokaryotes end up with new genetic material that they didn't have before. So they're both horizontal gene transfer, but they're not the same process, right? So the correct answer here was D. They're both horizontal gene transfer, but the source of material is different. So one of them is because it's transported by a virus, and the other one is just picked up in the environment. What's the other type of horizontal gene transfer that I haven't listed here?
Conjugation.
Conjugation, exactly. And conjugation, what happens?
Pili goes over, it's like whenever, something like that.
Yep, exactly. So you got these little pili that come out of the cell and they touch the neighbor, and then they transfer that piece of DNA. It's usually on plasmid across to the other cell. All right, so those are the three types of horizontal gene transfer that we talked about. Conjugation, transformation, transduction. Questions about any of that stuff? Okay. We already kind of talked about this. You guys want to, instead of answering this question, why don't you tell me 3 pieces of evidence that support mitochondria and chloroplasts once being their own independent organism?
I don't know these ones. I just don't.
Yeah,
They divide independently.
they do. They do divide independently within the cell. What do they also have in them? They have their own DNA, and it's circular in a plasmid-like structure, just like prokaryotic DNA is circular. What else?
Isn't they found?
You're close. They have a double membrane. So if you imagine that you had a happy little prokaryote, and you have a big thing that comes along and eats it, when the big thing comes along and eats it, generally the way cells engulf another cell, or how they eat other things, is they just engulf them with their own membrane. So when this finally comes on the inside, you end up with 2 membranes, one that came from the host and one that came from the symbiote. That make sense?
Wouldn't the host be the larger one?
Yeah.
This large one.
I put it there for the large one.
No, I mean like the whole.
Oh, well, this is the host membrane also, but when it engulfs this one.
Oh, okay, I see now.
Yeah,
I got you.
okay. I asked for 3, but I've got one more for what's another piece of evidence that if these were formerly prokaryotes.
Well, no, haven't
I
they?
said so.
Haven't they found?
Yes, you should always, always trust everything people tell you. No, actually, I was thinking it's because, they're about the same size as a typical prokaryote as well. So, all right. All right. So everyone try and do this yourself and then talk to your neighbors for the ones that you could figure out. Give me an example of an organism from each of the following groups.